So as most of you know, brain disorders are a leading cause of disability worldwide and actually affect tens of millions of patients. And unfortunately, we're pretty bad at treating them. So among these patients, a large proportion are resistant to any of our current therapies, whether medication, psychotherapy. So for example, in the case of major depression, there's 5 million treatment-resistant patients in the US alone. So based on the idea that these disorders are signs of abnormal activity and signals in the brain, an idea that arises is whether we can revolutionize therapies by actually trying to control or regulate these abnormal signal patterns. Indeed, there's been proof of concept demonstration showing that if as input, we apply deep electrical brain stimulation in the form of these biphasic pulse trains, depression symptoms may be alleviated in some patients. So the video I'm about to show you is a video of one such patient. So you can see this patient clearly suffers from symptoms of major depression. At Emory School of Medicine, she underwent stimulation, electrical stimulation, implantation and electrical stimulation. Actually, that was the electrode from which her brain was stimulated. And this is what she felt afterwards. So this, of course, is remarkable, but unfortunately, it doesn't happen in every patient. And indeed, there's been a lot of variability across patients, meaning we have a long way to go to make these treatments effective. So how do we think about doing so? Where do we go from here? One aspect that we focused on when we started thinking about this problem was the inter and intra-individual variability that are known to be prominent in these disorders. We know there's variability in symptoms, in neural representation, and even in response to therapy across and within individuals. So what if, instead of having a stimulation device that turns on in open loop and stays on, we actually guide how we stimulate the brain by the very neural activity that we're trying to control and by dynamic input-output model of how the brain responds to stimulation. Can we do better? Now, I don't have to tell this audience, as control engineers, we've been pretty good at building closed-loop control systems, such as autopilot. So what's the big deal? Why can't we do this for the brain? So unfortunately, of course, we didn't design the brain. It's an immensely complex system of unknown dynamics, and that introduces a huge machine learning challenge to build data-driven models of this complex system and to come up with data collection frameworks that can enable this modeling. So that is what my lab focuses on. We work at the interface of machine learning and neuroscience and a little bit of control to develop model-based closed-loop decoding and control systems for the brain and enable next-generation brain-machine interfaces and neurotechnologies for diverse neurological and neuropsychiatric disorders. So given that this is a pretty broad audience, what I thought I'll do is to introduce some of the challenges and the progress we've made up to now on many of these fronts, just to give you a broad overview of this domain. Okay, so what is this closed loop system that we envision? We envision a system that would take neural activity, decode from that in real time an underlying state of the brain, let's say mood symptom in depression, use that as feedback within a controller to adjust the therapy parameters, in this case, electrical stimulation, for example, frequency, amplitude, in order to take the brain to a target state. So this is our classical setup. But of course, this model-based system, as I've depicted here, is unrealized as of today. And there's multiple computational challenges we have to overcome to realize this system. The first challenge is that in order to build a decoder, what I need first is an encoding model that would tell us, okay, well, how is mood even represented in brain activity? But that is not enough, because even decoding mood perfectly will not tell me how to stimulate the brain to regulate mood symptoms. So what we need additionally are input-output models that would tell us the effect of stimulation input on brain activity and therefore on the underlying mood state. And finally, we have to put these models together to come up with a control strategy. So what I'm going to do is kind of show you the progress we've made on realizing each of these boxes. So first, decoding of mood when we started had not been shown. And we said, okay, to solve this problem, let's first collect the right data set. In collaboration with Eddie Chang, who's a neurosurgeon at UCSF, we recorded intracranial ECOG signals in epilepsy patients who have these electrodes implanted already for their standard monitoring. And this allowed us to record from a network of brain regions that are implicated in mood. And while they were in the hospital for about a week to 2 weeks, we also asked them to intermittently essentially report their mood using an iPad-based questionnaire, just asking them, well, how do you feel right now? So what this did was it gave us a sparse number of mood labels simultaneous with this continuous high-dimensional neural recording. So this was the data set based on which we had to train our decoders. Now, this was a difficult modeling and machine learning problem because we had to model this very distributed high-dimensional neural signal space, but do so with very sparse labels of mood. So we showed a few years back that indeed we can decode mood from intracranial brain activity. This is work by two of my students, Omid Sani and Yusha Yang. And the way we did this was essentially by developing a dimensionality reduction approach. So the idea was that we know these representations are distributed, so there's therefore likely redundancy in the representation. So we developed an approach that essentially found the smallest possible subnetwork within this larger network that was good enough for decoding. So that spatially reduced dimensionality. And then even within that smaller network, we did further dimensionality reduction by training a latent state dynamical model that described the neural activity in terms of a lower dimensional latent state. And then we related just that low dimensional state to mood to hopefully avoid overfitting and build generalizable models. Once you have the encoding, the decoder is straightforward. You just build a Kalman filter and a regression. So does this work? We tested it in 7 epilepsy patients. I'm showing you decoded versus measured mood. So the closer to 45-degree line, the better. And you can see that we're doing a pretty good job at the population level. And more importantly, in every single individual, we could significantly decode their mood variation, thus opening the door to personalized tracking of mood symptoms over time within an individual. Okay, so what are the networks that our decoder found was very important for mood prediction? What we found was that even though our modeling was individualized, across the patients, the decoder always picked consistent limbic regions that are also known from neuroimaging studies to be implicated in emotion regulation. This was important for us because we wanted to have some biological consistency and we wanted to see the robustness of the approach. More important than that, what we found was that despite again, individualized modeling, in 60% of the patients, we found this region of the brain, orbitofrontal cortex, to be the most mood predictive site. Now, this is interesting because in an independent study in collaboration with Eddie, we went ahead and stimulated there. And what we found was that stimulation there actually alleviated mood symptoms. So patient went from describing their feeling to be on the sad side or a little bit nervous to describing it as being calm, cool, and collected and feeling a lot better. So these two studies, one totally based on modeling and machine learning, one based on stimulation and trial and error, arrived at a common node in the network, orbitofrontal cortex, indicating its significance in emotion regulation. All right, so that's briefly how we're addressing the first challenge. But as I mentioned, that's not enough. What we need to do next is to describe how electrical stimulation would change neural activity and therefore the underlying mood state, essentially build this model of this box. Now, this is the classical input-output modeling, right? There's been beautiful work on biophysical models to do exactly this. But the problem is that these biophysical models are disease-specific. For example, for the cortical basal ganglia network in Parkinson's disease. They're very complex, and therefore their parameters are not fit to an individual's data. They're not data-driven models. They're essentially population-level models. And therefore, they may not translate to developing a controller that we are trying to build. There's also been models, data-driven, of the response of a specific neuron or one cortical column to stimulation. But what makes it difficult in our case is that we need to model the response across a distributed, large-scale, multi-regional brain network, do that in every individual, and do it during temporally varying stimulation, which happens in closed loop. So in a recent work, we actually took an alternative approach and developed data-driven brain network input-output modeling frameworks to try to approach this problem. And the model that we developed was essentially, again, this low-dimensional latent state dynamical system model. But what we did this time was to hypothesize that the effect of stimulation actually is reflected in this low-dimensional latent space, and through that, in neural activity and behavior, let's say mood. But the question is, how do we train this model? How do we collect informative data by stimulating the brain and recording the response? Now, of course, for this audience, you all know from system identification theory, not any input pattern would work. We need input patterns that would excite the system across the frequency spectrum so that we can model that input-output relationships across the spectrum. So we need white noise type inputs. But we're talking about brain stimulation. What does that even mean for brain stimulation? Where in the clinic, people apply these periodic biphasic pulse trains that are far from being white. So the idea here was that it's really the parameters of these pulse train that drive clinical outcomes, for example, frequency, amplitude. So what we have to do is to design novel waveforms that are wide spectrum in the parameter space. So this is our design. We start with this biphasic pulse that is charge balanced and safe clinically. Then we modulate its frequency and amplitude according to a binary coin flip. With 50% probability, we stay at one level. With 50%, we switch. And what we get is something we call a binary noise modulated electrical stimulation pattern. Now, we all know this is wide spectrum in the parameter space. It's clinically safe, and it's very flexible because I can use any pulse shape. So this is just a proposal. It's a theoretical proposal. How does this really work in the brain? Does this modeling paradigm even work in the brain? So a couple of years back, we started a collaboration with Bijan Pesaran's lab at NYU, now at Penn, who has a monkey neurophysiology lab. When we applied our framework in non-human primates, essentially, we applied our real waveform to the brain. At one site, we recorded local field potentials at multiple brain sites, and we fitted our model and looked at its feed-forward prediction. So this is all feed-forward prediction. And what we found was that indeed our models could predict how neural activity changes in response to a time-varying stimulation, and could do so across two animals, different stimulation sites, and so on. Another interesting observation here was that while we were stimulating in one region, the response was actually distributed across multiple brain regions. But the strength of the response was variable. Now, the question was why? And our hypothesis was that this likely has to do with at-rest functional connectivity between where you're stimulating and when you're recording. And in particular, as a measure to test this hypothesis, we used controllability, essentially network controllability. We took address data, we computed the energy needed at a given source node to modulate the activity at each individual target node. So I can compute this. And what we found was that indeed this functional controllability measure explained the variability during stimulation, meaning the more connected the stimulation site was to a given target region prior to stimulation, the more strongly that region responded during stimulation, thus confirming our hypothesis. So that's briefly how we're approaching the second challenge. Now, the idea is how do we put these together to come up with a control strategy? This is the step we're currently working on, but I'll give you some intuition how we're working on this. So the first idea is, of course, if we have an input-output model, then we know how changing the stimulation would change neural activity. Now, if I build a decoder, then I know how that change in neural activity would change mood. So together, if I can combine these models, and that's not trivial because the time scales are very, very different, then I can have a transfer function and then build a closed loop controller. Now, before getting there, of course, we have to confirm whether our models have sufficient accuracy to even try to go there. So what we did was we ran a simulation where we simulated a brain with enough model mismatch to emulate how much error we see in our actual data sets. And then we built just a classical, what we saw this morning, a classical linear quadratic Gaussian regulator to take a simulated mood state to a target level. And what we found was that both in the best case of our models and in the worst case of our models, our controllers could actually track this target, meaning our models have sufficient accuracy, and they did better than model-free approaches that turn simulation simply, or model-based approaches that turn simulation simply on and off, showing the potential in going in this direction. Okay. So far, I've kind of walked you through this entire loop, described some of the challenges we're dealing with and the approach we've taken. But in order to make these systems robust and accurate and to have them generalize across diverse disorders beyond, let's say, chronic pain or paralysis and other disorders, there's multiple additional machine learning challenges that we have to overcome, and I'll briefly describe these. The first challenge is that these neural signals that we record from the brain actually have a mixed representation and mixed dynamics. What do I exactly mean by that? If you think about an experiment in which you're recording some neural signals and you're measuring some behavior signals, let's forget about mood for the sake of the argument. Let's say I'm now interested in movement signals, right? The problem is, so we usually want to relate these together and decode one from the other. The problem is that the latent brain signals that give rise to our neural recording do not all relate to the behavior of interest that we are measuring. For example, if I'm thinking about arm movement, maybe some of the recordings are related to my emotion at the time of movement or how hungry or thirsty I am. So what we really want is to dissociate and model those shared latent states between behavior and neural signal, those shared dynamics, the green ones. The problem, though, is that our current approaches cannot directly give us those green states because they're unsupervised, trained with respect to behavior. So they may give us the red state that while existing neural signals actually are not behaviorally relevant and can actually mask or confound the extraction of those green states. So we developed a new algorithm called preferential subspace identification, or PSID, that can directly calculate those green states. The idea is, again, a linear latent state dynamical system model, but the unique capability provided now is that PSID can actually dissociate the elements within the latent state that are behaviorally relevant from the other elements, and do so with priority, meaning I can first learn and extract those green states without even worrying about those other states. And this prioritized learning actually is key to the accurate operation of this learning algorithm. So how does PSID work? I just give you a very brief overview. I'm not going to go into the detail, but the basic idea is that those green states should summarize everything we know about future behavior in terms of past neural activity. So in a first stage, PSID projects future behavior signals onto past neural signals, gets that prediction, and then from the row space of that, which we can get with an SVD, we prove that we can get the behaviorally relevant latent states. Once we have those, then if we want the additional states, we can now compute the residual neural activity that is unexplained so far, project this future onto past neural activity, do another SVD, compute the behaviorally irrelevant states. And once you extract the states, you can simply fit the model parameter. So that's just a basic description of the method. How does it work? We this time tested this in non-human primate data sets where monkeys were performing reaching and grasping movements to diverse locations. We were also recording neural activity from 100 channels or so. And the idea is, can we decode their movement from their brain activity? So this is how we can do. So I'm showing you PSID compared to, again, a linear dynamical system model trained unsupervised with respect to behavior. So the model structure, the decoder structure, everything is the same. The learning algorithm is what is different. And you can see that PSID better decodes movement at every single state dimension and can actually do this better decoding at a much lower state dimension, so 5 times lower. And the reason for that is, again, that prioritized learning. You extract the relevant states first, so you can describe them much more parsimoniously. And this actually holds even when we compared with nonlinear models like sequential autoencoders that are trained unsupervised. Okay, what is the next challenge? Everything I've shown you so far focuses on a single spatial temporal scale of brain activity or a single modality. But of course, we know behavior is represented across multiple spatial temporal scale that we can actually record for different sensors with different modalities. For example, at the small scale of single cells, so we have these intracortical arrays that record spiking of single neurons and local field potentials. Then we have at the larger scale these surface grids, we call them ECOG grids that record activity there. So the question is, can we go multi-scale with our models and do sensor fusion for the brain? Now, computationally, this is hard because these signals have very different statistical distribution and time scales. So spikes are these action potential events. So you can basically model them as a binary time series, whereas LFPs, ECOG, these are continuous, have slower time scale. So we've been developing multi-scale dynamical models that describe how a given latent brain state is represented across all of these modalities with a combination of Gaussian and point process observation models. Now, this is, of course, a new model structure. So we needed to learn the parameters by developing a new learning algorithm, a multi-scale learning algorithm. We had to develop a new decoder to aggregate information across discrete and continuous modalities. And then we could go multi-scale with our dynamic model. So I'm not going to go into the algorithms, just briefly. If you're curious, they're all published. The multi-scale filter essentially runs at multiple time scales, aggregates information. And you can think of it as a generalization of Kalman and point process filters that in the update step, you essentially update based on both modalities. And we use deterministic approximations to derive them. The learning algorithm is an expectation maximization algorithm, multi-scale version that iteratively maximizes the likelihood to find the parameters. But what I'm going to focus on instead, now that we have these, what interesting things can we do with them? So in a recent paper, we actually used this methodology to look at the relationship between small-scale spiking dynamics of single neurons and large-scale local field potential dynamics during this naturalistic reach and grasp movement that I described. Now, the good thing is once you fit that model, if you want to look at dynamics, you can ignore the two last lines and just focus on the first line. And in particular, the eigenvalues of that state transition matrix in general are these complex conjugate numbers. We call dynamical mode. Their radius tells us how fast neural dynamics decay in time, and their radius, and then their angle tells us with what frequency they oscillate over time. So essentially, they summarize the characteristics of your neural dynamics. So we looked at these for spike alone, for LFE alone, and for combination thereof using our novel methods. And what we found was that even though, of course, there were modes that were distinct between spike and LFE, there happened to be one mode that was showing up every time at a very similar location, meaning it had similar decay and oscillation characteristics. And then we said, okay, so what's up with this mode? What's special about it? So we looked at how well it decoded movement, and it was dominantly predictive of movement compared with the other modes. So this showed that this naturalistic movement actually engages large-scale brain networks and their coordinated action. And because this shared mode existed, we could actually aggregate spike and field modalities with our multi-scale decoder to infer and decode it more accurately. And because of that, we could decode movement much more accurately by aggregating these modalities compared to the single scale decoders. We are also doing various other extensions of this multi-scale framework, for example, assessing multi-scale causality and connectivity by extending the directed information context to multi-scale signals. We're developing multi-scale switching dynamical system models to take into account regime switches, but I don't have time to go through these. These are mostly published, so you can take a look if you're interested. Okay, what about the next challenge? So of course, if we want these brain-machine interfaces to work chronically over time, we have to worry about non-stationarity, adaptation, plasticity. Let me just visualize this for you. So I'm showing you one channel of ECOG from one patient 44 hours apart. But just looking at this, you can see dynamics are changing. So obviously, if I change my model's fix, I'm going to fail. So what we need is actually adaptive models that track and adapt to these non-stationarities. So we've been developing adaptive learning algorithms by incorporating essentially a forgetting factor or learning rate to update the parameters of our latent state dynamical system models. And we have ways of actually picking this learning rate also systematically. We can also do this in real time and efficiently as well. So we actually applied this in a human ECOG data set, and we found that this adaptive approach can predict the dynamics of ECOG more accurately compared to the non-adaptive approach. And it can also do further dimensionality reduction through this more accurate characterization, showing the potential for this adaptive methods for developing neurotechnology. So far, everything I've shown you has had one unifying property, and that is the fact that I kept the dynamics equation linear. Every time the state equation was linear. So why did I do that? Of course, I want to design a system. Linear dynamics are amenable to real-time inference, to closed-loop control. But there's, of course, nonlinearity in the brain. So how do we take that into account? So basically, what we want to do is to describe this relationship between neural activity and behavior. So we write this relationship in terms of 4 interpretable mappings. A mapping from neural activity to a low-dimensional latent state, the recurrence or dynamics of that state, and then the mapping back to behavior and to neural activity. Now, in a linear dynamical system model, all of these mappings are linear matrix multiplication. But what we wanted to do was to allow each of these mappings to be linear or nonlinear independently of each other. Because we don't only care about nonlinearity, but also localizing it and understanding something about the brain. So we wanted to have some degree of control over this. We also wanted to extend our framework to behaviors that are not necessarily continuous values. So for example, are categorical. We wanted to allow for behaviors that are not continuously measurable, so they could be measured at intermittent discrete times. And finally, to neural patterns that could be Poisson distributed, not necessarily continuous. So we've developed recurrent neural network architectures that meet all of these properties. The input to these RNNs are neural activity, and the outputs from these RNNs are predictions of neural activity and behavior. And the parameters each correspond to one of those interpretable mappings that I mentioned. So this is nice because now I can go in and manipulate these interpretable mappings. For example, I can keep them as a linear matrix multiplication, or I can swap them for a nonlinear mapping in the form of a multi-layer neural network. So this has those characteristics, but of course, how do we learn it? So we wanted to learn this in a manner that prioritizes, again, the learning of those behaviorally relevant latent states. So we developed a 4-stage optimization algorithm. In the first 2 steps, we first extract the behaviorally relevant dynamics and states and their mapping to neural activity and behavior by essentially optimizing the explanation of behavior from neural activity. And in the last 2 steps, we find the additional state, the additional residual dynamics by now optimizing the prediction of residual neural dynamics. So how does this work? We have all of the properties. We have nonlinearity. Because this is an optimization, we can have intermittent measurements into our sum. We can change the cost to have categorical behaviors of Poisson distributions and all of that. So how does this work? So we tested this on 4 independent data sets from 3 independent laboratories. These were doing arm movements, eye movements, cursor movements. And we looked at 3 independent neural modalities. So spiking activity, raw LFE, and LFE power features. And what we found by comparing this RNN PSID algorithm with normal linear PSID is that indeed decoding of behavior was significantly improved by incorporating nonlinearity, and did so with every modality. We also looked at the degree of nonlinearity across different modalities, and the modality that benefited the most was the raw LFP signal. Now, this is interesting for us as neuroscientists. Well, I'm kind of a neuroscientist now too, because traditionally in neuroscience, what we do is we use power bands from LFP. These are traditional nonlinear features we extract. But we showed that if you use this RNN modeling, you actually get a performance boost, meaning you automatically discover nonlinear features that are more predictive of behavior and can even do better than spiking activity. So this was better decoding of movements and behavior. We also did better prediction of neural activity by looking at the scatter. You can see that. So we do basically go to the frontier of this performance of decoding versus neural self-prediction in all of the data sets. So this shows you the benefit of this nonlinearity. Now, I alluded to the fact that I want to use this as a tool to localize the source of nonlinearity potential in a given brain system. Meaning I want to know which of these elements are the best descriptions of the nonlinearity. So to do this, what we can do is we can build a fully nonlinear model and then models with individual nonlinearities and compare them together to do hypothesis testing. So we went ahead and did this in our data sets. And what we found in this data set was that describing the nonlinearity just in the readout from the dynamics to behavior was sufficient to best explain our data. So we said, okay, let's go look at the other data sets. These are all cortical data sets. And they all had the same property. So if I just kept the mapping from the state to behavior nonlinear, it was largely sufficient for my description. So what is the interpretation of this? This may indicate that most of the nonlinear processing is actually happening downstream of the cortex, for example, in the brainstem or spinal cord, for those of you who are neuroscientists and who are here. This gives you testable hypotheses to then go and design your experiments and test this with multi-regional recordings. All right. So finally, the last challenge I want to talk about is geometry. So how do we incorporate geometry? What do I mean by that? In neuroscience, across many, many systems, motor system, olfactory system, auditory system, people do dimensional reduction. Let's say they apply PCA. And one pattern that emerges over and over again are these rotational patterns over time. So we looked at this and we said, okay, why is this? And one hypothesis that we had was that maybe this is because our neural signals are evolving on low dimensional manifolds that are not just the hyperplane, but rather nonlinear manifolds that include a hole or a loop. And that's why when we project them, we see these rotations. So we went ahead and said, okay, if that is the case, then I can build a dynamical system model that learns and takes this geometry into account explicitly. And maybe that's the way I can write the state equation in a simpler form to make it amenable to control and inference. So that was the big idea here. So it took us about 5 years to develop this method. So this is very challenging. One of my most talented PhD students work on this. I won't have time to go through the steps, but I'll give you just a general idea. So essentially what we have are these noisy observations. So first we apply topological data analysis to first confirm, is there a hole? Is there a multidimensional hole or not? Once we discover that, then we fit that main hole structure. And then once we fit it, we say, okay, let's increase complexity of the dynamics. So we grow the dimensionality around that hole by building essentially local coordinate system, what we call appending dimensions. So going, for example, from a loop to a band. Once we do that, we have the structure, the static structure. Then we have a machine learning algorithm that learns the dynamics on top of the static structure. And you can see that the method in simulation works well. So you can reconstruct. So how does this work in the brain? The data set we looked at was this monkey data set I've been showing you over and over again. So remarkably, what we found was that there indeed existed a loopy manifold in the neural space. And the movement of the neural trajectory on that loopy manifold matched the movement of the arm in the physical space. So I could just look at where my neural activity is on the neural manifold and know where the arm is in the physical space very efficiently. And this was very interesting to us. And we are interested in actually extending this to doing control on manifolds and so on. All right, so that's all the challenges I'm going to talk about. We are very interested in integration of these algorithms with implantable hardware. So actually, one of the things we always take into account is that our algorithms need to be real-time and low complexity to be implementable and one day translate and help patients. So to conclude, brain disorders are a leading cause of disability. We work at the interface of machine learning, neuroscience, and control to develop methods for decoding, modeling, and controlling of these activity patterns to develop next generation neurotechnologies and hopefully help these patients. This is my lab who does all the work. So I thank my students first and foremost, and also the funding sources. And thank you again for the invitation. It's a pleasure to answer your questions.